When the first mRNA vaccines were approved for use in the public during the height of the COVID-19 pandemic, researchers hailed them as the culmination of years of hard work that could open up a new vista in vaccination technology that could lead to the ability to protect against diseases once considered a near-death sentence. The husband and wife duo of Ugur Sahin and Ozlem Tureci, who co-founded German firm BionTech, collaborated with pharma giant Pfizer to create one of the two major mRNA COVID-19 vaccines currently on the market and have now announced major strides in cancer vaccine research. The pair says they are optimistic that a vaccine against cancer might now be possible before 2030. On Sunday, the scientists appeared on a BBC program to talk about their mission to enable the human immune system to learn how to seek out and destroy cancer cells that are possibly left behind after a tumor removal surgery.
mRNA acts as a blueprint and allows you to tell the body to produce the drug or the vaccine, which you would otherwise produce externally in bioreactors, for example. And when you use mRNA as a vaccine, what you basically do is that the mRNA is the blueprint for the wanted poster of the enemy. In this case, cancer antigens, which distinguish cancer cells from normal cells. And we first invested our research towards finding and defining those cancer antigens, which allow us to paint the wanted poster, and then we used mRNA to communicate them to the patient's immune system.
The goal that we have is can we use this vaccine approach, this individualized vaccine approach, to ensure that after surgery, directly after surgery, patients receive a personalized, individualized vaccine, and we induce an immune response so that the T cells in the body of the patient can screen the body for remaining tumor cells and ideally eliminate these tumor cells and thereby reduce or completely inhibit the metastatic relapses, which would come 2 or 3 years later.
Although several previously promising cancer treatments have fizzled out after tests reveal their lack of efficacy and/or effectiveness, the couple says they are highly confident that this mRNA vaccine platform holds the key to what BBC presenter Lauren Kensberg called, quote, the answer to cancer.
I don't think so, because everything we have learned about the immune system and about what we achieve with a cancer vaccine shows in principle the clear activity. We can induce those killer T cells, we can direct them. The question is how, with which type of other interventions do we need to combine this, and what else do we need to tweak in order to have at the end of the day really patients' cure, patients' survival.
COVID-19 vaccines were the first to use the mRNA platform, resulting in a spike of anti-vaccination sentiment, largely due to misinformation and misconceptions that the technology was new and poorly researched. The truth is that these vaccines were just the culmination of over 2 decades of research into how to use mRNA to give instructions to the immune system to fight off various interlopers, and the researchers say that their success in producing a safe, effective COVID-19 vaccine gave them the boost they needed to accelerate the pace of research on addressing cancer in the same manner.
What we have developed over decades in cancer, for cancer vaccine development, has been the tailwind for developing the Covid-19 vaccine, and now the Covid-19 vaccine and our experience in developing it gives back to our cancer work. We have learned how to better, faster manufacture vaccines. We have learned about, in a large number of people, how the immune system reacts towards mRNA. And not only we, also regulators have learned about mRNA vaccines and how to deal with them, so that this will definitely accelerate also our cancer vaccine.
BionTech hopes their treatment can work for bowel cancer, melanomas, and other cancer types, despite facing several obstacles. One of those challenges is coming up with a cancer vaccine that targets all cancer cells without hampering healthy cells.